does the Wuhan coronavirus.
That started in China.
Originated in Wuhan.
Coronavirus originated in a lab.
Leak from a lab.
A laboratory leak.
That lab leak hypothesis,
Accidental lab leak hypothesis of coronavirus has been circulating right after COVID-19 epidemic started. However, many, including scientists, politicians, journalists, have called it a conspiracy theory. Even Wikipedia page calls it a conspiracy theory. Temperature around this topic is really high, as many parties have a dog in a fight. Does this theory hold any ground, or shall we put it in a trash basket with Flat Earth conspiracy and the theory of lizard men ruling the world? Just before we jump into specific topics, I want to make few things straight. 1. Analysis will be based on scientific literature, which will be linked in the video description and also inserted as footnotes for more clarity. 2. This is a YouTube video, not a scientific publication in Nature, so sometimes bear with me for using language which humans can understand. Now, let's get into it. Possibility that SARS-CoV-2 escaped from the Wuhan lab is not as crazy as it might sound at the first glance. There have been many notorious lab leak cases of deadly pathogens, and let's look at some of them. First, we start in Birmingham, Great Britain, where on August 11, 1978, medical photographer Janet Parker suddenly became ill and was diagnosed with deadly disease, smallpox. At that time, due to worldwide vaccination, it was thought that smallpox was eradicated. So what happened? Well, Janet Parker was working in medical school of Birmingham University, where smallpox samples were studied. As later investigation concluded, it escaped from the lab due to ineffective containment, killing last person who died of this disease. Second case relates to flu, which broke out in 1977 in far east of the Soviet Union. Using early genetic tools available at the time, 1977 H1N1 virus causing epidemic was found to be closely related to influenza viruses circulating back in 1949-50. Explanation of natural origin of this time-traveling virus, which almost has not mutated for more than 20 years, was weak. After many years and decades of studies, the fact that this virus escaped from the Chinese lab and then spread across thousands of kilometers has been scientifically explained. Next up is SARS-CoV-2's cousin called SARS, which is also a type of coronavirus and caused epidemic outbreak starting in 2002. In July 2003, it was declared as contained by WHO. But in September of 2003, there was a lab leak in Singapore. Then there was lab infection case in December of 2003 in Taiwan. As if it wasn't enough, in 2004, SARS infected several individuals in a very secured CDC laboratory of Beijing, China. 1 person died, 8 got SARS positive, and hundreds were quarantined. So not just viruses, but specifically coronaviruses already have many successful escapes under their belt. To be fair, in all these cases, it was already known virus that leaked. SARS-CoV-2 is different because it has never been found in nature previously. So we need a closer look how it separates itself from other coronaviruses. Coronavirus SARS-CoV-2 causes disease COVID-19. Coronaviruses are a big family of viruses, including alpha and beta coronaviruses, which can infect mammals, including humans. From the known human coronaviruses, our arch-enemies are mostly concentrated in the beta coronaviruses and are divided into 4 lineages: A, B, C, and D. SARS-CoV-2 and its cousin SARS are in lineage B of beta coronaviruses. We will get to why lineage B is important very soon. Coronaviruses utilize some tricks to enter into the cell, and we will simplify this process for the video purposes. Invasion starts with attachment to a target cell, which then proceeds with fusion to the cell's wall, called membrane. For the attachment, SARS-CoV-2 uses host cell receptor called AC2. After the attachment, there comes fusion with the cell membrane. In order to start fusion process, tip on a spike of a coronavirus shall be removed. And this removal paradoxically is done by cell itself, using different enzymes. From wider range of enzymes virus can choose, more dangerous this virus could be. SARS-CoV-2 is unique because it can use enzyme called furin. On a spike of the SARS-CoV-2, there is a special place for this important cut, called furin cleavage site. What's profound is that none of the known representatives in the lineage B of beta coronaviruses have a furin cleavage site. Enzyme furin is widely expressed in human cells. It's in brain cells, endocrine tissues, kidneys, pancreas, and etc. So access to furin increases pathogenesis, and it even may be the crucial new feature causing emergence of SARS-CoV-2 in humans. Obviously, SARS-CoV-2 shall have some unique skills, otherwise it wouldn't have been so widespread in the first place. To explain this particular feature, some scientists have proposed that SARS-CoV-2 might have recombined with other viruses and acquired furin cleavage site.
This is a recombinant.
But this explanation is challenged in scientific literature, claiming that natural recombination is less likely and lab manipulation is more likely. Coronavirus's spike, used for cell attachment, is a very complex protein which consists of 1,273 amino acids sequenced in a specific way. This is a sequence of amino acids which makes a furin cleavage site, and for some scientists it looks really strange. For example, on this position, amino acid proline appears only in 5 furin sites out of known 132. Presence of amino acid alanine here is also unusual in furin cleavage site and appears again in only 5 out of 132 sequences. Now, if we take even larger chain of amino acids near furin cleavage site, this motif is similar to neurotoxins of cobras and kraits, and neurotoxin-like regions from the rabies virus. Is this similarity just coincidental, or is it more plausible to think that furin cleavage site was inserted in a lab? As of now, there is no 100% answer, but understanding some of the cutting-edge techniques in virology research will be useful. Virology involves experiments that can produce gained or lost functions in the viruses. For example, you can add special spike protein to a virus or cause a loss of the ability for a virus to replicate well. This type of research is called gain of function. It allows scientists to make predictions in which way viruses could evolve and even start preemptive research on vaccines before outbreak. One of the methods used by scientists in gain of function research is serial passaging, when virus is grown in a certain environment and then passed to a different environment. This could be done in tissues or living animals and puts extreme evolutionary pressure on viruses. Serial passage does not require engineering viruses from scratch, so it's a powerful and effective tool in the hands of scientists, but also dangerous, since there are risks of unexpected species cross-contamination, including infection of humans. There has been a big controversy whether we shall be doing gain-of-function experiments at all. Controversy got to new heights in 2012 when these two guys, Ron Fushier and Yoshihiro Kawaoka, increased transmissibility of deadly H5N1 influenza virus in mammals. Coronaviruses are no exception to gain-of-function research. Such experiments on bat-borne coronaviruses has been ongoing for nearly 2 decades. For example, in 2006, American virologists inserted furin cleavage site, interesting fella you already know, in SARS virus. SARS does not naturally have a furin cleavage site, and as we said, only SARS-CoV-2 in the lineage B of the beta coronaviruses has it. So insertion of furin cleavage site in coronaviruses is not a science fiction. Actually, as scientists have pointed out, inserting furin cleavage site in laboratory is possible without any evidence that there was a manipulation. Now question is, what role does China, and more specifically Wuhan, play in laboratory experiments on coronaviruses? Wuhan is, let's say, interesting place for coronavirus pandemics to start. With a population over 10 million people, Wuhan is a real hub of advanced bioindustry. City also has now infamous Institute of Virology, a world-class research facility. In 2018, Wuhan Institute of Virology opened first ever BSL-4 laboratory of China. What's a BSL-4 lab? Well, laboratories are ranked by biosafety level from lowest level of 1 to maximum level of 4, and there are only few dozen level 4 labs in the world. These special labs are used to study life-threatening diseases for which no vaccines or therapies are available. Wuhan Institute of Virology, with its Level 4 lab, in addition to some Level 3 labs, has intensely been working on coronavirus diagnostics, isolation, and vaccine development, well before 2019. One of the leading figures of this research is Xi Zhengli, who directs Center for Emerging Infectious Diseases at Wuhan Institute of Virology. For her groundbreaking work, in press she is also known by a nickname, Batwoman. With her colleagues, in 2013, Xi Zhengli discovered 2 new bat coronaviruses in Chinese horseshoe bats living in southern part of China. This discovery was unique because they found first wild-type coronaviruses of a bat origin which uses AC2 receptor for cell entry. Interestingly, this research and other later studies were funded by EcoHealth Alliance, where Peter Daszak is a president. Why this matters? Well, this is a person which was selected by WHO as one of the investigators of SARS-CoV-2 origin. Of course, during the selection process, it did not matter that he's buddies with virologists of Wuhan Institute, and they have drinks once in a while. You shall not then get surprised that, according to this guy, lab leak is much less likely scenario than possibility that SARS-CoV-2 was imported in China via frozen food from another country. To return to Batwoman, after 2013 she had a much bigger fish to fry. In 2015 her team announced that they generated a chimeric virus which had a spike protein from a bat coronavirus inserted into a mouse-adapted SARS virus backbone. What do we know so far? SARS-CoV-2 has a potent furin cleavage site which does not exist in its close relatives. Modern science tools allow enhancement of viruses in the labs, and Wuhan is one of the best places for coronavirus research. But does it mean that since scientists in Wuhan had capability to enhance coronaviruses and pandemic started in this city, SARS-CoV-2 leaked from the lab? It's like accusing person of murder when a dead body is found in his yard and in his basement he has largest collection of lethal poisons. You kind of get suspicion about the guy, but is it not just a pure speculation without further context? Now we need to look into this context. Usually viruses do not fall from space, and scientists nowadays have sophisticated tools to research their origin and spread. Peter Daszak might be drinking too much with Xi Zhengli, but he certainly knows virology. As he has stated in a scientific paper, rarely viruses gain ability to spread efficiently in a new host that was not previously exposed or susceptible to that virus. To the contrary of that, SARS-CoV-2 was perfectly adapted to infect humans from the get-go and had no apparent need for adaptive evolution at all. Somehow it already had a whole biological arsenal at its disposal. To undermine lab leak hypothesis, and in order to demonstrate that SARS-CoV-2 spread from natural origin, extremely similar viruses or progenitor viruses shall be identified. As WHO has stated, neither of the viruses identified so far in the mammalian species are sufficiently similar to SARS-CoV-2 to serve as its direct progenitor. To provide perspective how unusual this fact is, we shall compare SARS-CoV-2 to previous coronavirus pandemics. As Batwoman herself indicated in a paper, SARS in 2002 and MERS 10 years later were transmitted to humans via intermediary hosts. After several months of investigation in 2003, scientists identified SARS-like virus in Himalayan palm civets, which were traded in a live animal market of Guangdong, China. Evidence of infection was also detected in other animals, including a raccoon dog, poor rocket, and in humans working at the same food market. so animal market infection was easily detected. An identity rate between human SARS genome and that of the closest palm-civet virus was 99.52%. Similarly, researchers found out about camels having antibodies for the MERS pretty soon after the outbreak. MERS virus originated from the Egyptian tomb bat and was transmitted to a dromedary camel before going on to infect humans. Fundamental question is, where are SARS-CoV-2's natural progenitors with 99.5% similarity? There are other red flags too. Adaptation of a virus in humans should result in widespread outbreaks, not a single-point outbreak. There should be a lot of intermediary types of viruses between the progenitor virus and the current SARS-CoV-2, which we see no evidence of. Actually, we see absolutely opposite picture. WHO says that in an extensive exercise by 233 health institutions in Wuhan, 76,253 records of cases of respiratory conditions were scrutinized clinically for the October and November of 2019, just weeks before the outbreak. This testing determined that none, in fact, was due to SARS-CoV-2 infection. WHO report has been criticized for the lack of access to raw data. And of course, more studies are required. But this is best information which we have as of now. Serial passage of coronavirus in a lab can explain this discrepancy. Serial passage artificially condenses a natural phenomenon into a small temporal window. And this could potentially give us insight why we do not have any known intermediary pathogenic coronaviruses in nature prior to SARS-CoV-2. Coronaviruses can potentially spread either directly from bats or via intermediary hosts. Bats have a great immune system and can carry many kinds of viruses without becoming ill. There is a huge natural coronavirus pool in bats that sometimes can spread to humans. So thinking that humans got SARS-CoV-2 from bats has a scientific basis. However, when analyzing urban peculiarities of megacities, apart from Gotham of course, you do not think about thriving bat colonies or bat caves. When WHO conducted investigation, incomplete though, presence of SARS-CoV-2 has not been detected through sampling and testing of bats or wildlife across China. Testing of more than 1,100 bats in Hubei province, where Wuhan is located, has not shown any results close to SARS-CoV-2. We, people outside of China, have a misunderstanding that in all of the Chinese cities they are eating bats and selling them on food markets. This presumption about Wuhan is absolutely wrong. There was a very interesting pre-publication in February 2020, done by two Chinese specialists, one of which was working in Tianyu Hospital in Wuhan. This article argued that there is a low probability for bats to fly to Wuhan, especially its center. As the Chinese authors indicate, there were municipal reports, resident testimonies, and seafood visitor testimonies that bats were never a food source in Wuhan, and no bat was traded in a food market. Even without this paper, WHO investigation checked Wuhan seafood sales records for December 2019. There were no bat cells found there. As direct infection from bats is unlikely based on current evidence, maybe it was some intermediary host. SARS and MERS spread it through intermediary hosts, so it's plausible for SARS-CoV-2 to do the same, right? Many people thought that intermediary host origin is a very likely possibility because of Hunan market in Wuhan and large number of COVID-19 cases associated with it. Wuhan seafood market sells some wild or exotic animals, so this assumption is reasonable at first glance. Thing is, Wuhan seafood market, where many cases were initially discovered, is not source of SARS-CoV-2, and there is a strong scientific consensus on this. Despite search for viruses in the animal species sold on this market to date, no intermediary virus has been identified. 3 of the first 4 patients admitted to hospitals which got confirmed COVID-19 had no association or exposure to seafood market. Of course, search for intermediary hosts shall also extend outside of Wuhan. Ironically, if more tests are done and still nothing is found in nature or farms, this only increases chance of accidental lab leak hypothesis to be correct. It's also very strange how contagious SARS-CoV-2 is to so many mammals, and at the same time, there are no spreads of close relative viruses recorded in China until epidemic suddenly broke out in humans in December of 2019. In contrast to China, SARS-CoV-2 spread like a wildfire in European farms. In November 2020, European CDC published a report that Denmark, Italy, Netherlands, Spain, Sweden reported outbreak in mink farms. Denmark reported infected mink at more than 200 mink farms and had to kill more than 15 million animals. After the slaughter in Denmark, China, which was on second place, became world's leading mink fur exporter. Some researchers have suggested that culprit was a very rare animal, pangolin. This pangolin euphoria started because in March and August of 2019, Chinese customs conducted anti-smuggling operations in Guangdong and obtained illegally trafficked pangolins. It was reported that these animals were infected with coronavirus, and similarity of its genome to SARS-CoV-2 was 90.1%. However, there are several serious problems with pangolin origin hypothesis. First, pangolins have long been banned from sale and could not be seen in the market. Chances of their sale in Wuhan is extremely low. Also, 90% similarity of pangolin coronavirus with SARS-CoV-2 is very distant to speak of one being another's progenitor. Coronavirus from the direct intermediary host should be closer to humans than to bats, which is not the case here at all. These pangolin coronaviruses actually indicate that there is a huge genetic distance that needs decades of evolution. To put this into perspective, humans have 90% similar DNA to cats. So this kind of similarity does not lead us anywhere. Animal sampling and testing is the only way to establish a natural origin, be it in the wild, in the Chinese mink farms, or etc. However, instead of open and honest examination of these causes, we have seen only obstruction from Chinese authorities. For many months, China was blocking WHO team to enter Wuhan and study origins of SARS-CoV-2. Investigation team was allowed only in 2021, almost 2 years after the outbreak. Imagine police examining a crime scene 2 years after the crime was committed. So considering actions of Chinese authorities, one would argue, is also important. While searching for COVID-19 origins, there are some inexplicable, strange coincidences. You might say that they are trivial, but I want to provide these facts for your judgment. First is that China has a very important database of bat viruses, which could hold essential information on SARS-CoV-2 origins. For mysterious reasons, on September 12, 2019, just several weeks and months prior to pandemic outbreak, this base was turned off. Second strangeness is reported by WHO itself. Apart from BSL-4 lab in Wuhan Institute of Virology, there is a Wuhan CDC laboratory. It hosts animals for research purpose, specializes in pathogen collection and identification. Not only Wuhan CDC lab is less than 300 meters from Wuhan seafood market, where many cases happened in December of 2019, but CDC laboratory moved to this place on December 2nd, 2019, just when outbreak was starting. As WHO stated, such moves can be disruptive for operations of any laboratory, but there was no further analysis provided. Third strangeness is related to Batwoman, when in February of 2020, while COVID-19 pandemic was gradually taking over the world, she published a paper in journal Nature. This paper states that Wuhan Institute of Virology found 96.2% similarity of SARS-CoV-2 to a sample preserved in their lab named RATG13. This coronavirus was detected in a horseshoe bat, and to this day it remains the closest relative of SARS-CoV-2, which public has awareness of. However, this article failed to mention when, where, and how coronavirus RATG13 was found. Some scientists decided to dig into this. On November 17, 2020, after long delays, Segreto and Dagian published the article where they identified that RATG13 has 100% match with partially published genome of bat coronavirus 4991. But 4991 was found many years ago in 2012. And does this mean that full genome of RATG13 was unreported for 8 years? Simple answer is yes. On the exact same day, November 17, 2020, Batwoman's article in journal Nature gets addendum. She admits that RATG13 was indeed found in 2012. She also shares a story which international scientific community, or WHO, had no awareness of. According to this addendum, in April 2012, several miners visited a mine cave in Tongguan Town, Yunnan Province, to clean bat feces in order to mine copper. These miners got severe pneumonia and were admitted to hospital of Kunming Medical University. Batwoman and her team got samples from miners and then started research of that mine. Between 2012 and 2015, for several years, groups sampled bats and collected hundreds of samples. They detected highly diverse coronaviruses, of which 9 were beta coronaviruses. RATG13 was unpublished for 8 years without full sequence and origin backstory. Full sequence of other 8 beta coronaviruses has not been yet published, so we do not know was RATG13 really closest relative of SARS-CoV-2, or are there even closer matches which could serve as its laboratory progenitors? Answer to this question is crucial because scientists do not claim that RATG13 is SARS-CoV-2's direct progenitor. On the contrary, its genetic distance is still too high. Finally, to make things even more complicated, in April 2020, China started censoring research regarding origins of COVID-19. Publication of such articles requires government approval. Maybe this is just one of the ways authoritarian government wants to strengthen its position. Or maybe they know true story of origin of COVID-19 and want to bury it as deep as they can. If in 2020, reputation of a scientist could have been burned on a stake for stating that lab leak was possible, in 2021 it's no longer a heresy in scientific community. In April 2021, many well-respected virologists, biologists, and scientists issued open letter to WHO demanding to look into the lab leak hypothesis. After WHO published report in 2021, WHO Director General clearly stated that all possible origin theories are still on the table. Former head of CDC in the United States stated that he believed virus accidentally leaked from the lab. Let's make a quick summary of key facts. Lab leaks, including coronavirus leaks, have happened. SARS-CoV-2 has peculiar furin cleavage site not found in its close relatives, and its zoonotic origin is rather shaky. Virologists, including specialists in Wuhan, have capabilities to enhance coronaviruses. As of now, there is no evidence of SARS-CoV-2 evolving in nature, and it suddenly appears perfectly adapted for infecting humans right in Wuhan, which is a center for bat coronavirus research in China. No bats or intermediary hosts have been found with SARS-CoV-2's progenitor virus or sufficiently similar coronavirus. Seafood market of Wuhan was not place of origin of COVID-19. China has actively hidden information, removed databases, and obstructed WHO investigation. All these facts make accidental lab leak hypothesis a logical suggestion, deserving attention and analysis. purpose of this video was not to argue that there was an accidental lab leak, because science has not yet demonstrated this. Worse, it seems science may not be allowed to test this hypothesis. Video just points out how absurd it is to call accidental lab leak hypothesis a conspiracy theory. Despite our wishes, pandemics most certainly will happen, and even might happen in our lifetimes. But to prevent such a catastrophe, we shall not be afraid to search for the truth, and shall not silence them who want an open discussion about pandemic origins. Instead, we shall look truth in the eyes and follow it, wherever it leads us. Thanks for watching.